טוב, בוקר טוב לכולם. אנגלית, נכון? Good morning to everyone. My name is Udi Zohari. I will be teaching you the stuff on the visual system. And just to get an idea, how many of you heard me give lectures in the course Neural Mechanisms of Animal Behavior? Okay, 1, 2, 3. 1, 2. Okay, so there's going to be some overlap, although here I'm going to emphasize the computational aspect. So, but I need to speak a little about the biology, obviously. Forgive me, some of you, as I said, the first part is going to be sort of just an overview of the sort of biology of the visual system, which includes also computational aspects. So bear with me. And let's just get started. When we're talking about, as I said, I'll be teaching you about the visual system. I want to, first of all, emphasize, give you some sort of overview, because I understand I'm the first one who's giving you something about a sensory system. I think you've heard a sort of a general overview about sensory systems, right, from Elin Elkin in the first talk. And so I don't know if we got into this, but I always make the point of sort of telling you something about what is the essence of, what's so amazing about the neural computation that's involved in the visual system. And that is that if you think about this, what you have impinging on your retina. So I think many of you, if you've never heard a talk in the university about the visual system, know from high school that there's the issue of sort of the visual image being imprinted on your, the lens, and it's imprinted on your retina, and of course it's inverse. That's people learn in optics, in 10th grade or something like that. And then there's the problem, you have to invert again the visual image. That's what, when you talk to people who are in the street, that's the problem that they emphasize. And of course, that's not the problem at all, okay? The problem is how to make sense of what we see, because there's no, okay, you have to invert the image. But of course, what you have on the retina, if you think about this, is first of all an image impinging on the retina, and then this is transformed into electrical signals. And you could think about the electrical signals as sort of as you have sort of an array of photoreceptors. And here what you see is just these numbers which correspond to the level of activity. Let's say that there's a transformation between the light intensity and the level of activity of neurons in photoreceptor neurons in the retina. Okay, so you can think about these as sort of photoreceptor number 1, 2, 3, and they're organized in this array. And these numbers correspond to the levels of intensity of the light, which corresponds now to the activity, electrical activity of these photoreceptors. And of course, one has to take this electrical activity and make sense of this. And this is, of course, what's being done by the brain. Okay? There's some, and we'll talk about today, some computation already being done at the level of the retina, quite an amazing one. And that we'll focus on today. But most of it is done at higher levels in the brain, and I'm not sure if you're aware of this, but about 30% of our cortex, our cerebral cortex, is devoted to vision. Okay, and again, so you have this, and then you immediately transform this to an image here, which you interpret, right? And I'm sure you have never seen this image before, unless you've heard my talk at some other, okay? And this is an image of, I think this is in Sri Lanka. I'm not sure where exactly is it. That's not really what's the important thing. The important thing is that you see people here hanging on, the electricity cables, right? Okay? And the reason is that there's this, that's the famous flood from, I think, 2005 or something like that, that followed the great earthquake near Indonesia. And you see this, and although you've never seen anything like this, you immediately interpret this, what is happening, what next to expect, okay? And you understand this like that. And the amazing thing is that you somehow transform this into something that you interpret without having been taught how to do this. Okay, you've internally learned to interpret the world, and that's very different. I'm sure that, you know, being in LTech and reading the news, you read about these autonomous cars, and you know that presumably are able to do something like that. Seen understanding, okay? These cars are able to drive your car and to interpret what's happening on the road at every moment and moment, okay? So something very similar to that, right? But what you do not know, I believe most of you, is that it's true that we use very sophisticated statistical learning algorithms that you'll hear from Tali Tishby or from Weiss, or people from computer science. But the thing is that, and this uses an architecture somewhat similar to neural networks in some very basic aspects. I'm not going to get into this today. But the interesting thing is, okay, and they sort of change their weights so that you get such a picture, and you learn, and the autonomous car learns to figure out, okay, is there a horse coming? Is there a passenger trying to overpass you with a car or something like that? But this is it learned, as I said, what you do not know, maybe, is that this is learned because there's 200 or 300 computer software engineers in Sri Lanka or in India giving examples to the program, and hundreds and thousands of exemplars in which the program learns to interpret what is there, you know, by the street, okay? And once you have this information through statistical learning, it learns to interpret the world. But unlike this, and this is sort of, you give the exemplars and you annotate each object in the exemplar. You say, this is a cat, this is a horse, so on and so forth. This is a signpost, and so on and so forth. And after this information, you know, with this annotation, which is done by humans, okay, if there's enough information that's supplied to the system, it learns to interpret, you know, the road, what happens in the road quite efficiently, so that you are able to let it drive your car. However, we learn this internally, and that's another very interesting aspect. I'm just mentioning it to you because I'm interested in this, but it's not something I'll cover here. But I just want to make the issue of what is learned, and you know that this is a very sophisticated computation done automatically by our brain, and it's not a very simple one. Okay. And the way we think about this is when you think about visual image processing, that there's different levels of processing. Okay, you can think about the low-level vision, intermediate-level vision, and high-level vision, which is related to visual cognition. I'll give examples of this in a moment, and we can map roughly what areas are involved in low-level vision and intermediate-level vision and high-level vision. But I just want to give you what's being done again computationally, from a computational aspect. And you can, I mean, this is a typical way of thinking about this from a sort of modeling way of thinking, is that initially in low-level vision, what you have to do is basically extract the contours. Find out where are the sort of differences in luminance, which relate to contours. But contours can be extracted from many, many different cues. It can be from luminance, as I just explained to you. You know, you see a line here. It's a difference in luminance, in this case in chrominance. Okay, but it can also be from motion. Okay, so if something's moving, okay, sort of random dots that are moving in a random dot background, stationary random dot background, would immediately pop out as a sort of a contour which is moving. Okay, didn't supply you with an example, but it could be based on color, it could be based on texture, it could be based on various cues. Disparity also, which is a difference in depth. Okay, and so that leads to contour extraction, and we think about low-level vision as sort of culminating in boundary assessment. And then there's an intermediate level vision where there's other cues, such as shape from shading, okay, and occlusion, other global motion aspects, which lead you to the computation of 3D surfaces. So it's not just contour lines, it's what the surfaces are. And finally, we have high-level vision, which is basically the highest level in which you identify specific objects. And agents, I mean humans, actions, understanding social settings. So this is, the highest level of what we call visual cognition. And it serves vision for action. Of course, vision in itself is important, but, vision is mostly used for action, of course, and outcome prediction, as I said before. So this is sort of just a general overview. And it's important to notice that it's not only that we have sort of the input coming from the retina leading to low-level visual areas, going to intermediate-level vision and high-level vision, but there's also feedback, okay? So the feedback is also very important. Again, I don't think we'll have too much time to dwell on it, but it's, and there's a big question about what does the feedback serve. So we think about this as serving maybe attention and most likely also for learning, okay, to shape the way the analysis is done in the low-level and mid-level vision. Okay, and that's just sort of to give you an idea in terms of the visual system. Here I'm just drawing you what we'll see in the next lesson, not today, about cortical processing. Okay, so but that just shows you the cortical areas which are involved in vision. Okay, so there's like about 30 different cortical areas and where they are in the brain. That's the back of the brain. The eye would be here. Okay, and information comes from the retina via the thalamus to V1. And then what you see, the arrows correspond to connections between various areas. So as you see, there's like a plethora of different areas. And what I want you to notice that for each forward arrow, such as from V1 to V2, there's also a backward arrow, the red ones, which means the feedback. Okay, so as I said, there's just as much feedback as feed-forward processing in the visual system. And there's about, as I said, more than 30 areas in a macaque monkey. In humans, we don't really know, we don't have a final count, but it's at least as much as in the macaque monkey. Okay, so, and again, sort of just as an overview, I'll sort of focus today about early visual processing from the retina to V1, okay? And so the point that you can think about this is, okay, you get an image, okay, and as I mentioned, there's some processing that's being done already in the retina, and then the information is sort of conveyed to the lateral geniculate nucleus, the lateral geniculate body. And what you see here is basically sort of, okay, there's both color processing and sort of processing in black and white luminance already done in the retina. We'll get into that in a minute. And then there's something about information done already at the next stage, which is the sort of the LGN, the lateral geniculate body, which we'll mention it very briefly. And then information is propagated to the cortex, the visual cortex, V1. And what I showed you in the previous slide was, forward from V1, okay? So this is just now the information as it goes to primary visual cortex of V1. Okay, so that's just to give you a basic picture of the anatomy. Okay, and I want to emphasize another point. This is something I always start when I sort of in my first lecture, and brings up, I think, the visual system is the key example for people mentioning, well, look how sophisticated the eye is. Okay, how on earth do you expect this to evolve using natural selection? Okay, and I'm quoting Darwin in The Origin of Species to say that, okay, let me just read this and let's dwell on it for just 1 minute. If numerous gradations from a simple and an imperfect eye to one complex and perfect can be shown to exist, each grade being useful to its possessor, as it certainly is the case, if further the eye ever varies and the variations, okay, so you need selection and you need variation, and variation be inherited, as is likewise certainly the case, and if such variation should be useful, to any animal under changing conditions of life, then the difficulty of believing that the perfect and a complex eye could be formed by natural selection, though inseparable. Who's an English speaker here? Does anyone know what inseparable means? Okay, I thought myself to be an English speaker, but to tell the truth, I don't know whether. My guess is that it seems it's beyond our imagination, inseparable by our imagination, but I don't know what the word is. I always get to this answer one. I didn't get the answer yet, so I should look it up and Google it. Okay, should not be considered as subversive of the theory. And okay, so I want to just, since I think, thinking about evolution, if there's one thing that's important in biology, it's evolution. Everything else is just details, okay? Theodore Dobzhansky said this in the 1970s, so it's not, again, not my statement as well. But if you think about the eye as an exemplar, I just want to make the point that, okay, this is typically used in theological discussions, and therefore I think it's important to mention this. And the point to notice is that the eye has evolved, and you can find, what's amazing is that you can find that there are what's called living fossils, still living, which have the same eye as they had hundreds of millions of years ago. And the one example is, for instance, the Nautilus here, which evolved 540 million years ago. It's the same specimen as it was 500 million years ago, and it has what's called a pinhole eye. So what you can see here is sort of the evolution of the eye from just a region which is photosensitive, meaning that it can detect light. And you say, okay, what's good about detecting light? Well, if you can tell if light is a change in light, you can predict that something has changed in the world. Okay, maybe something to worry about, a predator or prey, right? So that obviously has an advantage, and this has evolved, as I said. You can find what's called the planarian cup eye spots in in the planaria, which is a very, very simple creature which has evolved something like 1 million years. I said 1,000 million years ago. Okay, and then you have sort of what's called a depressed folded area. And you see, basically, these are sort of steps of evolution. So if you look at the Nautilus, which is the Kicha, is sort of I don't remember what's the name in English, but anyway, it's sort of a marine animal living in depth. Okay, very similar to the octopus, but much older. Okay, then it has a very simple pinhole eye. So there's no lens, no cornea, nothing like that. But it's good enough. The pinhole allows finer direction selectivity. Okay, so you know something about which way is the object they're moving. Okay, and that obviously confers a great advantage. And the Nautilus lives in rather deep waters, so that's enough. Okay, there's not that much light there, and that's good enough for it. And it's still alive 540 million years later, and so on and so forth. So just to make the point here that through punctuated jumps in evolution, you could have evolved from a very, very simple sort of region which is just photosensitive to the fine eye that mammals and humans have. Okay? And here's just another example of the evolution of the eye with, from light-sensitive cells that's in one sort of marine see here different marine animals, and abalone, Nautilus, which I mentioned, so on and so forth, just to make the points. So I think this is sort of just for, from a general perspective, this is really important to know, okay? And I wanted you to be aware of it. Now, the other point I want to make sure is that some of the principles that we'll be talking about are very simple and can be found already in these very old living fossils. And one of them is the limulus, which is, again, from 300 million years ago. You can find if you walk in the sort of the shore of New England, near Boston and Cape Cod, you can find them. Easily dispersed on the, their shells in the sand. And the limulus has these eyes, okay? And it's called a compound eye, very similar to what you find in the fly, okay? And so here's a sort of a cross-section of the compound eye. And the point is, it doesn't have one lens as we do, but it has many, many lenses, each of them sort of focusing the light from a different point in space, okay? So you can think about this, instead of having one lens which focuses the light, here you have like a multitude of these lenses, and each of them has its own sort of photoreceptors which convert light from a specific position in space. Okay, so here's the sort of the array of these, the compound eye of the limulus, and there's sort of a cornea for each of them, and a lens, and never mind the details. What I want to make the point is that in the 1930s, this limulus eye was studied by Hartline. That's not, and basically, you'll see in a minute, a basic principle in sort of neural computation was found already at the limulus. I'll mention that briefly. It's called lateral inhibition, and we'll get to that in a minute. But I want to make also another point here, is that just for the comparison here, here's the vertebrate eye, okay? So here's the lens and the different, basically different levels of the retina, which we'll get into in a minute, has different layers here. And the point I make is that the photoreceptors, which are the one which transform the light, are actually embedded deep here. They're the innermost level in the retina. And light has to go through these different neuronal structures until it reaches the photoreceptors, okay? Not a great, it's not ideal in terms of an engineering, because you get some light scattering from these other cell structures here. You can see it here, okay? And so you can think about light here reaching the retina. The retina is back here. Here's the lens. And you see here these cell structures, which we'll get into in a minute, which are between the light and the layer of the photoreceptors, which is deep embedded in the pigment epithelium here, which is actually required to rejuvenate these photoreceptors. And therefore, this is not an ideal engineering structure. In the octopus, which evolved in parallel, it's not the case. You see the photoreceptors here, which are these bumps here, are actually the outermost layer. So light reaches them without having to go through these other levels or layers of cells. Now, another point to mention is that where our best resolution is the fovea. It's the 1 degree or so of visual field at the center of gaze. So the fovea is the area which we have the best resolution. And I'm not sure if you are probably aware of it at some level, but just think about it. When you read, you cannot read in the periphery. You have to move your eyes again and again and again to foveate the next word, which is about the size of a fovea, in order to resolve what the word is. Okay? So the fovea is your highest region of the best resolution. And there, if you can see, this is sort of stressed here, that the cells from the other layers are removed. Okay, they're removed, so there won't be any light scattering, and light reaches that area of the fovea directly. That's another reason why we have the best resolution at that area. Okay, so the point here is to say that, okay, first of all, some aspects have evolved in evolution along the tree of evolution in different ways. Okay, solved in different ways, and they're not always the ideal ways. Okay, but you're sort of committed to one direction, because here the photoreceptors are embedded in this pigment epithelium structure in the vertebrate retina, so that's how it's evolved there. Okay, and now I'm focusing. This is what I showed you. This is maybe a better slide to mention what I pointed out here is that the fovea here, you see there's what's called the foveal pit here. Pit, the בור כזה, and it means that in that area specifically there are the other layers are sort of moved to the side so light would reach it directly. Okay, so that's an important point to notice. And now when we talk about the fovea, I'll mention it very briefly, but I just want to make the point is that there's two types of photoreceptors in the fovea. You probably, who's never heard, am I just repeating something that you all know? What, is it new? To whom is it new? Okay, so one person is, okay, so I will be rushing now, okay, if it's only one person. But I'll just say that in the fovea there's two different cells. There's what's called the rods and the cones. And the cones are responsible for color perception, while the rods have a lower threshold, meaning that they're activated at night. You know, that they are activated when there's less, lower level of illumination. And you can see here, why are the cones responsible for color perception? Not because there's something special about them, but because there are three different types of cones. Okay? So you see here, this is what you see here. The x-axis here is the wavelength from something from 400 to 700 nanometers, which is the wavelength of electromagnetic. Well, we have spectral sensitivity in the electromagnetic radiation, and the y-axis here is the relative absorbance. So what is the chance that if you shoot a photo and it's sort of, it hits here, it goes through the lens, hits the retina, what are the chances of absorption? So it's relative. It means that, okay, there's a maximum here, and you normalize everything by the maximum. So you see, okay, one photoreceptor, what we call the blue cone, is most sensitive to 430 nanometer of light, blue light. But you see that it would also absorb a photon if it were in a different, it had a different energy because its wavelength was, say, 500 nanometer. But the chances are smaller. Okay, so that is reflected by the sort of this tuning curve here. Okay, so you see three Y tuning curves which correspond to three different photoreceptors, which are cone photoreceptors: the blue cone, the green cone, and the red cone. Okay. Well, there's only one type of rod photoreceptor, and that's its tuning curve. So it is most sensitive at green light. Okay, and now let's look at here. That's a very important one. So what you see here, the y-axis corresponds to the threshold light, the light intensity needed in order to activate that cone or that photoreceptor. Okay, and you notice a few things. Okay, so you take someone, you put him in a dark. And there's a period of dark adaptation. Okay, so initially, these photoreceptors need to change their gain so that be sensitive to much lower levels of illumination, fewer photons. Okay, and that takes a bit of time. But notice here that the dark adaptation gets to its minimum threshold level for the cones at a much higher level than the rods. What does that mean? Okay, so note two things. First of all, that for rods it's much lower, meaning what? Which are more sensitive, the cones or the rods? The rods, right? Because, okay, they reach the threshold for activating the rod is much lower than the cones, okay? So, indeed, at night, it's only the rods which are active, and the cones, if you reach a level of illumination below their threshold, are inactive. Okay? Point number one. Point number two is, okay, that if the illumination is, okay, just another point to notice, which I think you're all aware of, is that at night we do not see color. We may see grayscale, but we do not see color. Why? Because color is based on a comparison between the three types of cones, and maybe that's shown here. So let's think, how do you generate, and this is what's called the trichromatic color perception, which was basically invented by Young in the early 19th century, 1802. Okay, and Young noticed that you can. How did he realize this? Okay, at the time they didn't know anything about blue and green and red photoreceptors, which are active in daylight. But they did notice the following. They've noticed the following, that you can take and project on the screen light which is orange light at some level of intensity. Okay? And you can project on the screen a sort of a combination of both red light and green light. And here the radius corresponds to the intensity. Okay? So the red light is more intense than a green light. Well, here this is the intensity of the orange light. Okay? And what you can see here is, remember that I talked about the tuning curves of the different photoreceptors? So what you see here is basically the activation evoked by this orange light. So it would activate the green photoreceptor to some degree, indicated here by the green arrow here, and to a greater degree, the red one. Okay? This orange light is not ideal for either of them, but you see that there's a difference in terms of the level of activation you expect in the the red photoreceptors versus the green photoreceptors. Okay? Now, the interesting thing is that you can, if you now present on the screen, rather than this orange light, you present a sort of a mixture of some level of the green light and the red light, the perception is the same, orange. Okay? And why is that? Well, it's because if you think about it, okay, now you present red light. So now you would activate the red photoreceptor by the red arrow here, and the green photoreceptor very weakly, right? Which is indicated here by the small arrow here. But you also present the green light here. Now the green light here would activate both of them to the same degree, but since this is a weaker green light, then it's smaller in its scale. Okay? So the arrows here correspond to the degree of activation. Remember that the y-axis here is the relative absorbance. So we expect the absorbance of both the green and the red photoreceptor to be the same, but the arrow here corresponds to the degree of activation. So since you've given a weaker level of intensity, you expect a weaker degree of activation. But the point is that it's the same for the green photoreceptor as the red photoreceptor. Now what's important here is, okay, as I said, both evoke the same perception. How do we understand this? We understand this because if you sum the activity of the red photoreceptor, it's the sum of this arrow here, the red arrow here, caused by the red light, and the red arrow here, smaller one, but still a level of activation, caused by the green light, right? Because there's some activation due to the green light as well. Okay? So if you sum these two, you get this vector here. Again, a summary of this, a summation of this arrow and this arrow, which leads you to here. Exactly the same as if you were to present that orange light here, again activating the red photoreceptor at the same level of here. Okay? Same is true also for the green. Okay? So if you look at the green here, that is its level of activation, while the green here is the sum of this activation arrow here and this activation here, and they're the same. So if these levels of activation, the red, green, and in this case there's no activation of the blue photoreceptor, are the same, you perceive, Helmholtz and Young said that you would perceive the same color. Okay? And that's what's called color metameres. It means, okay, look here. So you can think about this as a sort of a relationship between the three photoreceptors, the activation level of the three photoreceptors. You have a vector which consists of three numbers, right? The blue, in this case 0, the green, and the red. And if this vector is equal to that vector, you perceive this to be the same light. Okay? That's the idea. Has everyone followed me? That's okay.
Question. I'm not sure about the right graph. Were they showing the two colors separately in measure or at the same time?
Yeah. Think about two projectors, okay? One with a red projector okay? Or one red laser, if you think about that, and the other one is a green one, okay? You know what the frequency that you're actually of this laser, okay? And now you ask, what is the perceived color? And you can change the intensity of these lasers to say, when are they the same, okay? And basically, this is called, as I said, this was sort of done. They speculated this. They didn't have lasers or anything like this, but this has been proven later, okay? And the point here is that color perception is based on this difference or this ratio between the activation of these three photoreceptor types, and this also induces this aspect of metamers, that you can have two different spectral profiles that can give rise to the same perception of color, as I said, just as in the example that I've given you. So you see here, you have just to mention here, what's important here is the level of activation of the blue, the green, and the red. And if they're the same, if these two vectors match, then you would perceive the color to be the same, although the spectra may be different. Okay? So that's an important point. When I'm talking about the spectra, let's just make it clear that it's understood to everyone when we're saying here that the spectral profile can be very different, leading to the same color perception. Think about it. It's more complicated than that. I won't get into it. I just want to seed the idea in your mind. I'm talking to you here, not sitting, but standing, talking to here. And this shirt here looks blue, right? Now, this is mainly from light due to fluorescent light. Now, I walk out, okay? Now the spectra of the sun is much different, is very different from the spectra of the fluorescent light. But you still appreciate my color to be blue, okay? So although it's called color constancy, although the spectral profile may change dramatically, you still perceive the color to be the same. And that's even more complicated than what I explained here. This is very basic, but there's another level that I just mentioned here. It's called color constancy, which we won't get into, which is sort of a computation that's being done not at the level of the retina, but higher level, probably in cortex V4 and so on and so forth. Okay, so that's something that I mentioned about color, because there's the different photoreceptors in the retina which are encoding color, but I want also to give you something about the structure of the retina in very, very short time, and then we'll move on to discuss other aspects. So I talked briefly about the cones and the rods, and remember I mentioned to you that there's sort of the anatomy is such that light comes, from sort of the back here, crossing these different layers here. So sort of you can think about the retina if you sort of expand this. Here's the ganglion cells, light coming here. They had to pass through these layers here, and to be transformed at the level of the photoreceptors, which are deep here, embedded in the pigment epithelium. But let's talk a little about the structure here, okay? So a few things. Okay, so light is converted here, and it has to sort of somehow pass through these bipolar cells and horizontal and amacrine cells, which is sort of in the inner nuclear layer here, to the level of ganglion cells, these cells are the ones which fire spikes, okay? So ganglion cells are the ones which send their action potentials out to the optic nerve and to the brain, okay? So these are the output cells, if you want, and these are sort of the ones who are doing the sort of the initial light conversion sort of and basic computations that are done at the level of the bipolar and horizontal cells. Okay. A few things to mention, first of all, is when you talk about the photoreceptor layer, as I mentioned, what you see here is the photoreceptor type, and the fovea is only cones. It's rod-free, okay? So here you see these are the rods, bigger, and these are the cones. So the cones are the one which convert, remember, they're the ones who convert color. Basically, they're responsible for color, and they have, in the fovea, their density is very, very great, leading to the best, as we said, best resolution. So resolution is to a great extent dependent on the density of the photoreceptor array. So you can see that this is the y-axis here, photoreceptor density, and you see that, okay, the cones drop sharply as you move from the center of gaze out. Okay, so the x-axis here is the distance from the foveal center. So this would be 0 would be center of gaze. And here you're talking about in millimeters as you move in the eye in millimeters, okay? And you can convert this, of course, to visual angles. So as I said, our best resolution is at the center of gaze at 1 degree or so of central visual field. And you see here the sharp decline of the photoreceptor, the cone photoreceptors as you move from the fovea out. And you see for the rods, you see that this is sort of the inverse picture. There's no rods at the center, and it sort of increases, and then it drops as you're moving to further and further to the periphery. Okay, another point to notice here is this. This is what's called the blind spot, which is sort of the area covered by the optic nerve in which there's no photoreceptors. Okay, so this corresponds to the blind spot. I'm sure you've all heard of this. If you've never had this, please come to me in the... Okay, no, don't come to me. Try this out on your own time. Okay, have a fixation cross and an object maybe some 10 or 15 centimeters away from your fixation point at maybe this distance. Okay, don't make it so big, make it something like this size, okay? And then close the right eye and move this back and forth, keeping fixation on your fixation point, and you'll see that the blind spot is the area in which you would not be able to recognize this object, okay? It will sort of suddenly fade away because it's now on the area, so on that area in which there's the nerves of the optic nerve. So there's no photoreceptors, and the light is not being converted into electrical signals from that area. Okay.
And when the blind spot is in place of the place where we create the optic nerve?
Yes, so the blind spot you can think about, okay, you have cables, as I said, that result from the ganglion cells, which have to transfer the information to the brain. Now these cables have to come out from somewhere, right? So where they come out is the area, okay, so you can think about this area here is the area in which there's no photoreceptors, leading to the fact that you'll have a blind spot in that area, okay? And now, so I've very briefly mentioned something about what's happening in the retina in terms of its anatomy, something about the color vision, and we'll later talk about the information as it goes from the retina to what's called the thalamus, or the LGN, which is a nucleus inside the thalamus, and then from this from the thalamus to V1. Okay, so here's sort of a magnification of the picture we see here, and the thalamus is divided into 6 different levels. So basically you talk about 2 layers, which are the magnocellular layers, and 4 layers, which are the parvo. Never mind the details, really. But the point to make is the following. I'm sorry, you know, the problem here is that I'm rushing, okay? Because I know a lot of you know this, heard of this, and then I'm somewhat worried that it's sort of, I may be boring some of you, and the others who have never heard this will have to read some, because I am rushing, will have to read. I'm just making some very basic points, and so you will have to, I've given some background information, but there's a lot of it in the paragraphs I've said that you should sort of cover. And so if you are unable to follow me, that's because of that. Okay, but basically, okay, when we talk about vision, the other thing to be aware of is that we have two eyes, okay? Again, that's for the exercise I told you here. You have to close one eye, okay? Because the blind spot of one eye is an area which is not the blind spot of the second eye, okay? So if you are not closing the other eye, you will be able to see the smiley because it does not fall in the blind spot of the other eye. And that's actually sort of portrayed here. So you think about this, there's the, both eyes, let's say, fixate here at the center here. And what is portrayed here is the fact that, you know, most of our visual field is within the binocular zone. But if you close one eye, I can't see what's going on there as I close one eye, of course, because it's outside, it's in the monocular zone, which is only mapped into one of the eyes, okay? So if you have a point, you fixate here and a point of light is presented here, it falls on both retinae of the left eye and the right eye, okay? And the point to mention here is that this information from the right visual field is transformed to the left hemisphere, and from the left visual field is transformed to the right hemisphere, such that the information from the, let's say, from the right visual field, when it hits the left eye, stays in the same field, and that from the right eye crosses what's called the chiasma here and moves to the opposite hemifield. Okay, hemisphere, not hemifield. Sorry, it's okay. And when Hubel and Wiesel started to study this, they've sort of had these sort of gadgets, very simple in the sense of what we have now, 50 years, 60 years after that. Basically what they had is they did their first studies on kittens, anesthetized kittens, in which they flashed stimuli and just recorded the electrical activity using sort of an oscilloscope after passing the signals to an amplifier, okay? So you could see the action potentials on the oscilloscope. And again, due to the lack of time, we want to take a break. And okay, so just to mention this very briefly before we take the break, I think you all know this, that we have, if you look already at the level of neurons, ganglion cells at the retina, what we find is that they have an area which is called an on-center area, or an area which they are, if you place light on the center area, you'd get great activation. If you increase the light to the periphery, you get a shutting down of the neuron, okay? So this is what's called an on-center receptor field. You have the opposite effect, what's called an off-center ganglion cell, which prefers light in the periphery and no light at the center, okay? We will talk about what does that mean in terms of computation. How can we understand this computation? So all this was the physiology. We'll get into the computation right after we take a break, and let's get back at 10. Okay, so sorry for taking 2 minutes of your time, but we'll continue. אפשר בעברית, רק שנייה, אפשר גם בחוץ. יש ספירה בעשר? בסדר, אז אנחנו לא נדבר על ספירה, אבל זה הספירה, אנחנו נמשיך. Okay, so there's going to be... כולם הבינו או רק זה? לא. Okay. There's going to be a siren at 10 a.m. sharp. It's due to the Memorial Day for the Holocaust. Don't be worried about it. We'll start immediately after that. Okay?
דבר ראשון, ההרצאות מועלות ליוטיוב ומועלות, נכון?
על היוטיוב, כן, אני חושב שכן, אני מניח שכן, אבל אני לא... א' הן נמצאות במודל. יש את המודל 2, וכאילו...
המודל לא ראיתי את זה גם. לא משנה, זה אני שואלת.
לא, כן, אני עכשיו מדבר מהמודל. הסליידים האלה העליתי אותם מהמודל לפני ה...
לא, המצגת כן, אבל ההקלטה כן. יכולה להיות הקלטה.
עכשיו מקליטים אותנו, נכון? מקליטים אותי עכשיו.
כן, זה אמור, כאילו, מתי אמר לי שזה אמור לעבור.
והיה אמור להיות שנה שעברה, אני לא יודע, אני לא מטפל בזה.
כן, לא, זה, אני אמרתי, אני שואלת דבר כזה.
בטח, כן.
אוקיי, אני רציתי לשאול, שדיברת על הגרפים האלה של הוקטורים.
בגלל זה אני שואלת, כאילו ראיתי אותך ואני חושית
אמורים להיות
זה שאלות שאתה צריך לקרוא את המאמר
אני כבר הייתי בזה, אם זה הנחיה של האוניברסיטה או אני לא...
זהו, זאת הבעיה. אחרי, אני אעשה את זה אחרי כאילו, מתי, Wow, I was really shocked about it. I was like, what the hell?
Nothing to do, you get rid of it.
Yeah, I know. Do I need to kick your ass out?
You do.
Okay. Everyone can hear me? Good. So we were talking about the properties of ganglion cells. Remember, these are the cells which project their output from the retina. I didn't mention at all what's happening in the photoreceptors. I'll get to this in a moment. But as I said, there's two classical types of ganglion cells. There's what's called the on-center and the off-center. And the on-center is the one which is best activated by light in its center of area and dark in its periphery. Now, there's one thing that's not mentioned here, but a key concept which I will reiterate, you probably heard this, it's called the receptive field. Okay? So what's plotted here is basically the receptive field of the cell, and it's in retinal coordinates, meaning that now if you have what I showed here, what's shown in the slide is basically an area in the visual field. If you fixate here, in which the cell, let's say, prefers light, which I will mark here by pluses, in the center and dark in the periphery. Now, if you move the center of gaze from here to here, then this receptive field moves together because it's in retinal coordinates to its new position, which is defined by this vector here, if you want, in terms of distance in x and y from the center of gaze. Okay, so this would be the receptive. It's the new receptive field, and it would still be responding to light at its center and dark in the periphery. And as basically shown here, this is just a schema for this. And of course, there's this other cell, which is the opposite, what's called the off-center ganglion cell, which prefers light at the periphery, at this annulus in the periphery, and that's basically shown here. I'm sure all of you have seen this, so I'm just jumping through this, but I want to mention something about the neural circuits and something about, remember that we had just the very basics of the sort of transformation of information from the level of the photoreceptors to the ganglion cells, which project their output to the brain. A few points here is, first of all, that the photoreceptors are surprisingly, if they basically transduce light, what happens is that they go through a slight hyperpolarization. So you see this is sort of the blip that is caused by transformation of light into an electrical signal at the level of the cone. And then this photoreceptor is connected to these bipolar, so let's look at the photoreceptor. It's connected to the bipolar cell here. But the synapse is inhibitory, so that in the bipolar cell, it's actually this hyperpolarization at the level of the photoreceptor leads to a depolarization at the level of the on-center bipolar cell, okay? Due to the fact that here the synapse is inhibitory, okay? And here the synapse is excitatory, so you get the same sign, okay? So this is, again, a hyperpolarization due to light, which is transformed by the photoreceptor here. At the next level, these bipolar cells are connected to ganglion cells, and notice that here again, there's no spikes. Okay, the level at which spikes occur is at the on-center ganglion cells, and if enough on-center bipolar cells converge on this on-center ganglion cells, then you get spike at the level of the ganglion cell here. And okay, so if you present light here, okay, at this area, if enough of these photoreceptors are activated, information is transmitted to this bipolar cells, leading to activation of these on-center ganglion cells. Okay, and I won't get into the full details of this. There's this aspect here, what's called a disinhibition here. See that light causes a hyperpolarization, and due to the hyperpolarization, there's less of a release of the neurotransmitter, in this case a glutamate, and then there's what's called a disinhibition. So this is sort of an inversion of the sign here from hyperpolarization to depolarization at the level of the bipolar cells. Okay. And just to make the point is that this connection is not one-to-one, but in fact, if you talk about a sort of a ganglion cell here and the bipolar cells, there's sort of a convergence of input of many, many cones into these bipolars, and from the bipolars there's also a conversion into the ganglion cell. So there's a great conversion. We're not talking about a one-to-one relationship, but tends to one in terms of the convergence at the level of the ganglion cell. Okay, and the other point, remember I mentioned in the limulus, this is physiology again. In a minute we'll talk about computation, okay? But just last point about the physiology. Remember that I talked about in the limulus that there's these different lenses corresponding to different positions in the visual field, right? And we'll mention this in a moment. What I want to, and I mentioned the point that in the limits already in the 30s, Kafar Hartline showed that there's this principle of lateral inhibition that we can find also in vertebrates. Okay, so a very, very basic principle that has evolved in evolution in the different pathways. Okay, and the idea here is how do we understand the fact that if you present light not only at the center but also at the periphery of this on-center cell, you get less of firing. Remember this? Okay, just to remind you. If you present light, diffuse, I didn't go over this, but if you present diffuse light covering both the center area and the surround, you get less of a firing of the cell. Okay, so you see, compare this to the ideal light conditions in which there's only light at the center, no light at the periphery, you get high firing rate. Here you get a much lower firing rate. This is just corresponding to the spontaneous firing rate of the cell. So how do you understand this? Well, the way you understand this is through lateral inhibition. And lateral inhibition means that, okay, that there's these connections which are done through the horizontal cells here, which connect photoreceptors in the periphery to photoreceptors in the fovea, okay, or in the center of the receptor field. So think about this is the receptor field of the cell, the ganglion cell, okay? Now remember, there's many, many photoreceptors. Each has sort of responding to light in different positions, okay, in this area, and together they all converge into this lateral into this to this ganglion cell here, through this sort of matrix here, lead to the firing of this ganglion cells, which is what we're looking at. Okay, the on-center and off-center is the firing of the ganglion cells here, and we're trying to understand this. And now what I explained to you before, very briefly, because I didn't have much time to get into that, is that, okay, if you present light at the center, activating these cones, okay, you get through the on-center bipolar cells activation of the ganglion cell as well. So you present light here, you get firing of these of this ganglion cell here. Now, what if you present light in the periphery? If you present light in the periphery, you should get less firing, right? You get inhibition. How do you get that? Well, you get this inhibition through lateral connection. That's why it's called lateral inhibition. So if you present light both at the center and the surround, you get less firing. Why is that? Because now you activate photoreceptors which correspond to that area in the visual field. Now, okay, you activate this photoreceptor. As I said, you always get hyperpolarization of this photoreceptor. Okay, but due to the horizontal connections here, okay, this horizontal cell, there's an excitatory synapse here, so this also horizontal cell is now hyperpolarized, but it connects one photoreceptor with a neighboring photoreceptor. Not immediately neighboring, but this may be a photoreceptor sort of mapping this area and a photoreceptor mapping that area. Okay, and what you find is that it actually causes depolarization. This connection causes depolarization of this photoreceptor, the neighboring photoreceptor. Okay? Now, depolarization is opposite to what you get normally, right? When you convert light, you get hyperpolarization. So now you get, because there's an inhibitory synapse here, you get a reversal so that there's a depolarization of the photoreceptor here. And this causes, if you remember the pathway that goes from photoreceptor to the on-center bipolar cell, there's sort of an inhibitory synapse here, so there's again an inversion of the sign, meaning that you get an inhibition of the on-center bipolar cell. And the result is that this on-center bipolar cell is connected to ganglion cell, and there's less firing of that ganglion cell. Okay? So think about, whoops, sorry. So think about this scheme here. Now, when you have activated both the center and the surround, you've activated photoreceptors, which are the on-center through the activation here, but you've also activated other on-center bipolar cells, which map the surround here, and they have the opposite effect, okay? So that you get less firing from the cells when you activate, when you use light both at the center and the surround. So here's how you explain through this very simple mechanism, lateral inhibition, why you would get less firing if you present light both at one position and another position, which is exactly what has been done by Hartline in the 1930s. What he did was, first of all, okay, so you have these, remember, the compound now, you can now project light to one of this, through one of these lenses here, activating these photoreceptors here, okay? So if you present light at point A, here's what they got. They got 53 spikes, and this is sort of giving you sort of the firing rate of the ganglion cell of A alone. Now, if you present B alone, this is what you see here, okay? Just to give you an idea, this is a time scale here, okay? So the x-axis is time scale. This is like at the day where they had to mark time, but you know, every 1.5 seconds they had a point here, okay? So you think about this as sort of just a piece of paper. This is marking time here, and this is the blip here is a spike that they record, okay? So notice now if you count how many spikes you had in this period of time, you had 46 spikes. How many spikes have you had if you had light at A alone? You had 53 spikes. Now if you present light at both A and B, what you get? Lateral inhibition. How can we see this? That this firing here is less than this one, and the number of spikes that you get here is less than this one. Okay, so they inhibit each other due to the mechanism that I've just explained to you very briefly. And this is just plotted here. Okay, so you can see that decrease, the y-axis here would be the decrease in frequency that you get, okay, when you activate both fibers. And you see here that, you know, there's as you increase the light so that fiber A fires more, you get more inhibition, a greater decrease of activity in fiber B, and the same here, okay? If you increase the light at fiber B, you get greater inhibition on fiber A, okay? So that's a sort of, it's nice to see that you see something that's already, you know, shown at the 1930s. Hartline got a Nobel Prize for, not just for this, he looked at the dynamics of this. It's much more complicated than this. I don't have time to get into this. But we now understand the circuitry that can explain this. Okay, so all of this was sort of basic physiology. I've been covering briefly what I show in this other course that I'm presenting this. And now I want to get into the computational aspects, which I think none of you, or probably none of you have heard. Okay, so let's think about it this way. Remember we talked about the retina as being an array, and you can think about it this as an array of photoreceptors, in this case the cone, mostly cone photoreceptors here at the fovea. So you see each one of them corresponding to light from different position in the visual field, okay? And now, what does it do? Okay, so you have this, so think about this. Now you have an image. It's been shown. The light passes through the lens and is now focused on the retina. It falls on the retina. And remember, I started, the very first picture I showed you was the retinal array. We're back to it now. Okay, so here's the retinal array. And now, remember, I talked to you briefly now about what these if you think about the ganglion cells, which are sending their out from the retina to the thalamus and from there to the brain, what does their filtering property look like? It's a center surround sort of function, right? So you can think about this center surround function as here's the receptor field structure. That's what I showed you, right? So it's either on center as shown here or off center, which is just the opposite. And you can think about this in terms of if you do a cross section here, it's just easier way to think about this. It's sort of you get something which would look like that, right? A function which would look like that, right? Activation at the center here, and deactivation or less activation, in this case hyperpolarization, or less activation than a spontaneous activity if you present light in the periphery, right? Now this is often called, the receptor field structure is called difference of Gaussians, or what's called Laplacian of Gaussian. Let me explain in a minute, okay? So difference of Gaussians is simple, okay? And it's often called dog or difference of Gaussians. Why? Because you can think about, we need light now. Okay, so you can think about, okay, just to think about the one-dimensional case, because it's easier to understand, you get something which looks like that, right? And you can think about this as sort of there's a Gaussian of excitation, which would, let me draw this in a different color just to make it clear. So there's a Gaussian shape of excitation, which looks like something like that, and a Gaussian shape of inhibition, which looks something like that, right? So the red is inhibition, that's why it's negative. The blue, the black is excitation, that's why it's positive. And if you just sum, you take this plus this, okay? You get the difference of Gaussian, which is the blue, function which I have shown here, okay? But another way to think about this, how many of you have a physics background? None. Okay, some physics background. Is to think about this as a second derivative operator. Let me explain. Okay, you have an image, okay? And let's, before we talk about an image, let's think about just something which is sort of a one-dimensional case. Okay, so let's say that you have something which looks like something, I don't know, just something like that. Just drawing. This is illumination as a function of x, of position. So if you were to, okay, so illumination is increasing. This would be light here and dark here, and it's changing in sort of in some fashion here. Okay, now you can think about a first derivative operator. What is the first derivative? Looking at where the change in light is maximal. Where would that be? So here's the light. This is a function. Okay, I is the intensity, and here x is position. And where would be the maximal change in light intensity? Right here, right? Okay, so you think about the derivative. Okay, so what do you have to do? You take the derivative, right? Okay, what is the derivative? Derivative is taking the difference between one point and another point here. Okay, so you're taking the difference in the illumination level. Okay, and so basically it's delta of I, right? When divided by delta x, as delta x is approaching 0, right? That's the definition of a first derivative. So if you were to take the derivative, it would look like, I don't know, something like that, increasing here, right? That would be the first derivative. Okay? Now you can think about a first derivative in space. What is it? It's taking the value that you have here, okay, and decreasing from it the value of intensity that you have here, this one, and decreasing from it the value of intensity that you have in a neighboring point, right? That's delta I, okay? Okay, that's the difference here, right? So that would be corresponding to an operator which looks like that. Meaning, you take the value at this point here, you give it a weight of 1, and you take the value at the neighboring point, and you give it a value of -1. And you take the difference between the two. Okay? So that would correspond to a first derivative. Clear to people here? Why is that the first derivative? Okay, what's the second derivative? What's the second derivative? Let's think about here, okay? So you have to, where would the second derivative, how does the second derivative look? You have to sort of look at the changes of the first derivative, right? So you look where the changes in the first derivative are mapped. You basically take this function and look at its derivative, right? So here, that would be 0 here, right? So it's basically, here you're looking at the changes in the slope here. Here the slope would be positive. Here the slope would be negative. And this would be the 0 crossing, right? A change in derivative from the second derivative from a positive value to a negative value. Correct? What is it? It's taking this and applying the same operator of the derivative. So I'm saying that this is an operator of a first derivative here. That's a first derivative. What would be an operator of a second derivative? Applying an operator, that operator to that again. So what would it correspond to? It would look like the following. Okay? So what you do is you move an operator which looks like that on top of that. So as you move it, if this is positive and this is negative, what would be the value? +1 times -1 is -1, right? I drew this opposite way. Is -1. When they correspond, you get -1 times -1. If they correspond, meaning that they're one on top of the other. The next stage is when they're one on top of the other. What would it be? It would be -1 here +2, right? Because let me explain in a minute. I'll just show you what it looks like. And -1 again. -1 +2 and -1. And that's a second derivative operation. Why is it a second derivative operation? Okay, because you slide the first derivative operator on your function here. And you look at the overlap between them. That's what's called the convolution. I'll show you in a minute what it means. I'm just giving you the sense of this. So when you slide it first, think about the red operator here. So it looks like something which is, this one is on top of this one. There's no overlap as I've shown here. So it looks something, the red looks like something like that. And then, therefore, here it's -1 times 0, meaning nothing. Then -1 times +1, meaning -1, which you get here. And then it's -0 times +1, again 0. Okay? So basically, what you get is an operator. This is the second derivative operator. Okay? And now, meaning that, okay, if I wasn't very clear to you, let's understand what the meaning of this was. Okay? So what I'm saying is, remember, we started with the receptive field properties of a cell. We said the receptive field property of a cell, the ganglion cells, are center-surround. They're organized in center-surround, and you can see them as either a difference of Gaussian, or you can see them as an operator, a second derivative operator. Meaning that what? That you're looking for changes in the luminance corresponding to the second derivative, in the luminance profile. Okay? So here's the luminance profile. I was talking, here's what the first derivative looked like. And if you look at the second derivative, it would look something like that. Okay, so if you take this at home. If you didn't understand anything of what I said, okay, take this and do the first derivative, you get this one, right? You do the second derivative, you get that kind of function here, okay? I'm saying that that is what is being done by the filters that we see at the level of the retina, okay? Just a second derivative, because that would be an operation of a second derivative on our image, which in this case is just a change in the luminance drawn here. Okay? And now the second derivative function is what's shown here. It has a negative area, a positive area, and again a negative area. But there's another twist to it, and that is, okay, so you can think of this this is just a second derivative operator, which is this, and it's sort of it's smoothed by a Gaussian. Okay, so if you do convolve this with a Gaussian, what you get is something which looks like that, sort of. Okay, and that's what's shown here. Okay, so it's a second, basically it's a second derivative operator that you smooth a bit, and you smooth it by a Gaussian kernel. Okay, so this is what you get. Now, if you didn't understand anything, let's see what happens to an image when you apply this. Okay, so here's, before we get into this, this is just what I explained to you in a minute, right? So here's your intensity edge a minute ago. If you apply the first derivative, this is what you get. If you apply the second derivative, this is what you get. Okay, so this is your, you can think about this. This is your image in one dimension, something moving from something dark to something light. And this is the first derivative, here's the second derivative, and the z here corresponds to the zero crossing, meaning that it changes from the positive lobe to the negative lobe. And what I'm saying is that basically what the filtering properties of the ganglion cells are doing is just a second derivative of the image. Okay, so it's taking the second derivative of the image. And I'm saying this, but the one who basically came up with this idea is David Marr. And have you heard of David Marr? You probably heard about him in sort of the sort of computational approach to vision specifically, but he did this not just in vision. So he came up with this idea that you basically, you can think about this from a computational aspect, that what the first initial analysis done at the level of the retina is extract this, basically perform a second derivative of the image, extracting the zero crossings. Okay, now the way to think about this is, okay, it comes from the what's called the convolution, and I'll try to explain to you without getting into the math, what do I mean by a convolution. So you think about this as the following. I just explained to you that, okay, you can think about each cell as a filter, right? And it has this difference of Gaussians or second derivative property, okay? And now, and remember, there's this array of ganglion cells which correspond to different positions in the visual field, each of them performing exactly the same operation. And the operation is this second derivative, or you can just draw it, okay? It's that sort of function, okay? Now so you can think about this as the kernel, which is now being operated on the visual image, okay? And so you have a function of what the visual image is. Let's say it's just what I drew before. Say, okay, something which x, intensity as a function of x, it may look something like that. Let's call this f of x. And then you have this being g of x, okay? So that's the function, that's the filtering property that you apply to each position in space, okay? And that's called a convolution, okay? So you're performing a convolution by multiplying, okay, at each position of space, the function of what the image is times the filter, okay? And summing up. Now let's give an example of what it is, okay? What do I mean by that? Maybe there's an example here. Okay, that's just, okay, before I give you a concrete example, just to give you a feel for this, okay, here's your image. Here's what the operation of the second derivative, you know, okay, so what you see here is a second derivative multiplied by a Gaussian. Remember, you don't just do a second derivative, just to remind you, but this is smooth somewhat, okay? So you can see this, okay, here's sort of, remember, this is the excitatory Gaussian minus the inhibitory Gaussian, which is the difference of Gaussians, but you can also understand this as a second derivative smoothed by a Gaussian.
Have I lost you completely, or are you following what I'm saying? Basically, the idea of a second derivative is you understood. What does it lead to? Okay, so what does it lead to? Here's your image. Here's how you apply after you apply the second derivative. So that would be sort of the activation profile that you would be seeing in your retina. Okay, so let's say that the darker it is, the lower activation, the higher it is, the lighter it is, higher activation. Okay, so this is sort of, you can think about this as sort of the activation profile that you're going to get from your retinal ganglion cells given an image. Okay, so that's a much more complicated image than I've showed you before, but it gives you an idea, okay, what is this convolution? What's the effect of this convolution? And just to explain, okay, so let's think about, just to make it very simple, let's think about two functions. Let's say that fx looks like this. Or the image looks like something like this, dark, light in a very specific region, and then dark again, okay? And let's say that our filter, rather than being this difference of Gaussian or second derivative, it's just a very simple filter, exactly the same, okay? Something like that. Okay, so that's Gx, that's our filter that we apply, okay? Now, what is the result of a convolution? If this was the image, that's a one-dimensional case, just to make it simple, and this is your convolution kernel, okay? This is the function that you apply, the filter that you apply. So let's think about this, just to make it, so here this is one-dimensional, okay? So what you do is basically, you do a sort of an integral of fx over gx, where you change alpha here, which is from minus infinity to plus infinity. Okay, nice math. Let's understand the basics of this. Okay, so what you do is you slide gx over fx. So you take this, and now you say, okay, here's gx and fx. What would be the result of the convolution for that point, x here? It's the multiplication of gx times fx. So let's say gx is 1 here, 0 everyone else, and here fx is 1 at this position here, 0 everything else. Okay, so what is the multiplication in this condition? The result of the multiplication. If you multiply this times this, it's 0 here except 1 in that area, but it's 0 here. So what would be the result? 0, right? Because 0 times 1 here, and here it's 1, but there it's 0 here. So that would be 0 at that point here. Okay, so I'm drawing here. Can you see in the back here? I'm drawing the result here. Now you move this. You start moving this. So you slide gx over fx. Okay, you move to the next step. Okay, so as long as there's no overlap between gx and fx, what are you going to get? 0. Okay, and now you start to get overlap. So it's 0. That's the result of a convolution. It's 0 here. Then you get to the area where there's starting to be an overlap. So the greater the overlap, the greater your value is, and it will grow linearly, okay, until they completely overlap one over the other. And then gx is moving further forward. Then the overlap starts to decrease, right? So you're going to get a decrease in the convolution, in the value of the convolution, and you're going to see something like that, right? Okay, now let's say, just to give a feel for this, what if fx was much bigger? Something like that. So here's the filter, this is the image now. So what are you going to get? Okay, so gx gets into here, and then it slides inside here, so you're going to get something. Can someone give me the result? Yes, so, yeah, so it would start, there's no overlap. Now the overlap increases. Now it stays at the same level until gx gets to this point here, right? This is gx now. And now it's sliding further on, and you get a drop here. Okay, you got the point. You got a feel for what this very sophisticated thing is. What do I mean when I'm saying convolution of the image by a specific filter? And what I showed you, whoops, what I'm showing you here is exactly the same thing. The function is a little more, the filter is this one. It's two-dimensional. This is just the one-dimensional case. It's two-dimensional, meaning that there's a center and a surround. And there's the image. It's a little more complicated than what I explained to you, but it's the same idea. You apply this everywhere. And why? Because the reason for this is because at every position in space, you have the same filtering property. Okay, so that's why you can slide one on top of the other. And here's what you get as the result. So now, if you lost me in the second derivative, it's good that you will understand this, but what's most important is that you understand now how did I derive this output, and you get a feel for what convolution integral does. So that is something that we understand. Okay, so now.
This stage of filtering is still in the retina?
Yes, these are the ganglion cells. Remember, the photoreceptors are just detectors of light. So the stronger the light, the more they respond. But this center surround structure is something that's keen to ganglion cells. Okay, that's the ones who are sending the output. Okay, and they're already performing. That's what I was trying to explain. That they're already performing a computation. And the computation is basically transforming such an image to such an image. Now, it's good that you mentioned this because what do we get in this image that was not that sort of emphasized with respect to this image here? What it emphasizes is contours, right? So basically, what you get here is where's a change, okay, a change in contrast, okay, between one region and another, okay. So here, of course, if you're changing from light area to dark area, so you get a great response here, okay? So it's emphasizing contours, okay? That's the intuition of it, okay? Now there's another point that Mar already that also mentioned, and by the way, okay, so if you want to get into this, you're really interested in this, I would advise you to have a look at David Marr's book from 1982 called Vision, where he developed these ideas. David Marr died at the age of 35, and he wrote this, the book was written after he died, okay, it was published after he died, but he made some really important advances in vision, in computational aspects. Some of them are outdated now, but I think that it's well worth reading. Okay, so, and I'm just giving you the gist of it, okay? And so, I mentioned that you get an edge detection in the images, so you can think about this. Okay, so here's your image now, and here's the operation that you perform, okay, so that you transform the image to this one, and then you can think about these zero crossings. Remember, the zero crossings is a change in the second derivative from a positive area to a negative area, and these are a plot of the zero crossings here, okay? So you see the sketch here is of the zero crossings, and they convey very well where the edges are. Okay, so David Marr made the point of mentioning that. And then there's another thing to notice is that you can have, here's your image, and you can convolve it with basically a filter in which this Gaussian is wide or this Gaussian is narrow. Meaning what? Remember that I was talking about a filter which looks like something like, in 2 dimensions, it looks like that. Okay, center surround. It could be this one, or it could be this one. They're both doing a center surround operation, but this one, of course, is on a wider scale, and this one is on a narrower scale. Okay, so this is reflected in this size of the Gaussian that you apply. Okay, so you can think about this as sort of, this is a map at the coarse scale, and that's a map of the fine scale. Now the edges, interestingly, are apparent both at the coarse scale and the fine scale. Okay, and then this is sort of portrayed here. So you can think about, and Marr mentioned this, that you can take the image and perform the same operation, perform the same filter, but once you use sort of a filter, a center surround filter, which is rather narrow, and once it's at intermediate scale, and once it's in a large scale. Okay, so the spatial scales change here. What's the consequence of this? Well, that's what's shown here. So if you look at the image, here's the zero crossing, or the contours that you can extract at the fine scale, and here's at the coarse scale, here's at the intermediate scale. It's very interesting, because what do you see? You see that, okay, the most important features are preserved when you do this operation at a very coarse scale here, okay? But you see that there's also very fine sort of level, which is contours which appear here and do not appear here, okay? So at multiple scales, you may get to finer resolution, if you want to say. Now, a comparison at the coarse grain is simpler computationally, or so often the way we think about this, I'm not getting into this, but just to explain a principle which was actually applied in computer vision. And that is, okay, you have an image, and let's say you have two images. I don't know if I have no, I don't have an example of this. And you have to compare them to say, okay, let's say for motion or for stereopsis. So how do you do this? Well, first compare them and look at the edges at a very coarse scale. Then you get some idea what corresponds to the other. Because to do a correspondence between one image and another image is very tricky. It's computationally very expensive. So do this at a coarse scale, get sort of a mapping at the coarse level, and then focus in on the higher, on the fine scale to do the fine adjustments. So that's actually a classical way that's being done. It's called pyramids. I may have time to mention this in my next lecture, but that's just for you to know that, okay, it has some bearing from a computational perspective. And that's just to make the point that, okay, we can understand very basic phenomena of perception just from that very simple operation done already at the level of the retina. And the classical one is the following, okay, which one of these looks darker, so this looks darker than this one, right? And so lightness depends, or perceived lightness depends on sort of the background illumination. And the way we can understand it, it's another example is here what's called Mach bands, is the fact, again, what you have here sort of is something that's changing in intensity from something which is bright here and changing gradually here, and here it's dark. And you still see here something which is a bright band here. Why do you see that? Well, you see this because if you apply the convolution, okay, so you have, in this case, you see a sort of a jump in the light intensity, and you apply a convolution, what you get is something which looks like, the output of the convolution is something that looks like that. And why is that? Well, think about, ganglion cell whose receptive field is here. Well, the area which is corresponding to the on-center area and the off-center are basically canceling one another, so you won't get much of a response here. As you're moving closer to the boundary, what do you see? You get, this is more optimal than this, in the sense that now some of the periphery, which should not have light, is now actually not lit. So therefore this, in B, you're going to have more activation than in A. If you move to C, the next step, then here this is the worst case in the sense, or suboptimal in the sense that there's dark covering all of the on-center area, and there's some light in the periphery here, which should be, the ideal would be that this would be dark here in the periphery, and there's only going to be light at the center here, right? So the result of this is a drop in the activation. So C is worse than D, because D, there's just dark both in the center and the surround. So D is not, you're going to get very little activation in D. In A, you won't get much activation because there's light both in the center and the periphery. But in B, you're going to get more activation than in A. And in C, you're going to get less activation than D. And that's basically reflected in the profile here. And this is the result that you can expect from a convolution of, you take a convolution of, this, and you convolve it with a second derivative function that I mentioned to you, you would get that sort of profile. And that, I failed to mention, but okay, that can explain the fact that here we see a light band here. And in fact, I didn't mention, but you also see a dark band here, right? And that is, so you understand the basic phenomena that we perceive just from the operation that's taking place at the level of the retina already. Okay, now I've passed my time. In the next lesson, we're going to talk about some operations that are being done at the level of V1 already beyond this, and we'll talk about some analysis done in terms of edge extraction versus an analysis done at the Fourier domain. Okay, so at the level of spectral analysis. And more to come on Sunday about this. Okay, להתראות. בטח שאפשר.
קודם כל, ממה שהראית, אני כבר נתקלתי בזה בעבר, אבל פתאום זה גם עכשיו, בעצם
מה?
אנחנו מתחילים עם הפונקציה.
שם תוצרה, או עם הנגזרת שנייה. זה היה, כאילו, זאת אומרת שאת באת
אני מתמטיקה בעיקרון, עשיתי גם חסות מפיזיקה. אבל בסיסיים, כאילו מכניקה, יחסות.
לא, זה היה מעל הראש או שזה היה ברור?
לא, הרעיון היה ברור. הפונקציה זה כאילו מפסיד איזושהי.
כן. לא, לכן
לא, אבל אז
עם הקונבולוציה, נכון, זה היה כאילו קל להבין, נכון?
כן, אז מה שאני אומרת, פתאום חשבתי, הפואנטה היא שאנחנו, הריאקציה שלנו היא פשוט לשינוי בכמות הפרוטונים כאילו שמגיעים לעין.
התגובה שלנו זה שינוי במיקום, כן? זאת אומרת, כאילו במקום אחד לעומת מקום שני. זאת אומרת, הדבר, כן, אבל אני כבר רוצה להתאמה. כאילו הדבר הקלאסי זה באמת שהוא הדגשה של רגישות אופס. כן, אני חושב שהדרך טובה זה לחשוב על זה. הנה, זה, התמונה הזאת. זאת אומרת, אוקיי, יש לך את התמונה. מה האוטפוט? התגובה של התאים של הרטינול גנגליון, זה מה שזה יראה. פשוט כל אחד מהם מתייחס למיקום אחר.
הם חייבים להיות כאילו ביחד, שזה באמת כאילו ביחד בתאים העון ועוף.
פה דיברתי רק, זה נכון שפה דיברתי רק על העון, העוף הם עושים את הפעולה הפוכה. אז נגיד פה הפס השחור הוא של העון, אז של העוף זה יהיה בדיוק ההפוך. נכון? זה יהיה פס לבן.
ואיפה שהראית את זה אחר כך עם הגנגליונים, אז הם היו גם עוף וגם עון, נכון?
נכון,
היו רק עונים.
אז פה התייחסתי, כל הזמן דיברתי רק על עון, כאילו, על התגובה כאילו של העון ריספונס לבד. עכשיו צריכה להגיד בעצם, העוף עושה את התמונת ראי של זה.
כן.
אוקיי, אז תקבלי תמונת ראי מבחינת
לא, אבל תעביר עוד אחד? עוד אחד קדימה כאילו, לא אחורה?
כן, אז זה...
עוד קדימה?
כן, וזה הקוסמוס שהזכרתי.
הנה, לא, עוד קדימה, סליחה. וואו. הנה.
זה און.
אז כולם און, אוקיי.
כן, רואה, פלוס במרכז, מינוס ב...
כן, חשבתי רגע שהם היו חלקות. בסדר גמור, ושאני לא אשכח, מה שהכי רציתי לשאול אותך, זה בשיעור קודם, דיברת על
בשיעור קודם, לא, אני הגעתי לך. בשיעור היום
כן,
בשעה הראשונה.
בשעה הראשונה. אז בכלל היה את הגרפים בקשר לקורן, שיש את שאמרנו שכאילו זה 3 וקטורים, ואם המספרים זהים, לא משנה איך קיבלתי את זה, אני מקבלת אותו פרספשן כאילו. כן, זה. אז, ואז, אחרי זה אמרת...
הסקאלה קונסטנסי.
שמה? לא, אחרי זה אמרת, כאילו, הנה תראו, עכשיו החולצה שלי כחולה, באופן מסוים, ואני אתן החוצה, כאילו... אמרתי, כאילו, יש עוד רמה, סיבכתי. אני מצטער. אמרתי שיש עוד רמה יותר מתוחכמת. זהו, זה לא אותו דבר.
זה לא אותו דבר, אני מצטער. זה רמה יותר מתוחכמת, כי פה...
זה כבר הידע שלי, כאילו.
לא, כן, אז כבר מה שואלת, אז אני אסביר במילה אחת. כי פה אני מדבר על זה כנראה ככה. בעצם מה שחשוב זה האוטפוט של 3 הוקטורים האלה. אתה מסתכל על 3 הוקטורים האלה, האוטפוט הזה שווה, מבחינתך זה אותו סדר. עכשיו, יש עוד רמה יותר מתוחכמת. זאת אומרת, אני מסתכלת על, כמו שאמרתי, על הצבע של החולצה שלי, באור של הפלורסנט ובאור של השמש, הספקטרום של האור, של שמש, הוא שונה לגמרי מספקטרום של האור של פלורסנט. יחד עם זאת, כן, מבחינת התפיסה, זו אותו תפיסה של צבע.
אבל זה בגלל הידע הקודם שלי.
לא, זה לא, אפשר להסתכל על זה ככה, לא, זה בגלל שאת אומרת, מה שחשוב, מה שלא משתנה, זה תכונות הרפלקטנס, תכונות ההחזרה של החומר. תכונות ההחזרה של פונקציות ההחזרה של חומר. זאת אומרת, רק הגעתי. לא דיברתי על זה, אז כאילו, עכשיו את שואלת משהו שאני קצת יותר מתוחכם. זאת אומרת, בעצם, אבל את יכולה עכשיו להבין את זה לאורך ההמשך של ההרצאה שלה. אני אומר, האוטפוט יהיה, בעצם יש לך פונקציה שמתארת, זה זה הספקטרום של האור. כן, זה הספקטרום של, אם את רוצה, האינפוט. אוקיי? אז הוא כאילו מ-400 עד 700 ננומטר. עכשיו יש את הספקטרום של הפילטרינג פרופרטיז. עכשיו, כאילו, על גבי זה את מפעילה את fx, שזה הפילטר שלה. אוקיי? והפילטר שלה, הוא נראה משהו שהוא, יש לו איזה, אני חושב רגע, כי יש את העניין של הרפלקטנס, אני מנסה עכשיו רגע איפה נכנס לעניין של הרפלקטנס. וואי, אז חכי רגע, אני לא בטוח עוד מתמטיקה איך להציג את זה. שנייה.
לא משנה.
כן, כן, זה גם לא מה שאת רוצה, לא, כאילו אני אומר, התכונות ההחזרה של החומר, הם לא משתנים, כן, זה בעצם הפילטרינג פרופרטיז, זה כאילו חומר...
החומר עצמו.
כן, החומר עצמו...
הצבעים של החולצה.
כן, שכאילו הם קובעים את הצבע. הצבע נקבע לפי החומר, אוקיי?
נכון.
והם לא משתנים.
ההחזרה שלו את האור.
בדיוק, אז הפונקציית ההחזרה היא לא תלויה אם זה באור אחד או באור שני. וזה מה שאנחנו יודעים לתפוס. זאת אומרת, זה בדיוק העניין. זאת אומרת, כאילו אנחנו...
אבל זה לא עושה לי שכל, כי נכון שזה מה שזה לא...
למרות שאת אומרת כאילו, אוקיי, כאילו, זה בדיוק אני רוצה לדעת.
אבל אני מכירה את העולם לפי מה שמגיע לי לעין. אז זה לא משנה אם הדבר עצמו הוא the same, אני מקבלת משהו אחר.
לגמרי צודקת. זה מה שאומר שבעצם אנחנו יודעים להבחין בצבע בגלל שאת אומרת, אוקיי, תכונות ההחזרה הן לא משתנות. כל הצבעים, התכונות ההחזרה שלהם הן קבועות, אוקיי? ולכן את מהירה באור מסוים או באור אחר, אתה לומד, אם את נותנת רק צבע אחד, אז לך קשה לזהות אותו. אבל כיוון שכולם משתנים באותה צורה, עם אור.
אתה לומד את הפונקציה הזאת.
בדיוק. זה מה שנקרא לקונסטנסי. אתה יודע...
זהו, זה מה שהתכוונתי.
נכון, וזה ברמה אחרת לגמרי מפה. זה הרבה יותר מתוחכם ממה שאני...
כן, אבל מה שמגיע לי לעין זה משהו אחר.
מה? כן, כי זה ברור שזה תלוי, כאילו מה שמגיע לך בעין זה מכפלה של, זה מה שהתחלתי לעשות לך פה, המכפלה של האור שהוא יכול להיות, אם הוא בספקטרום אחד או בספקטרום שונה, כן, אז זה ברור שזה ישפיע לך, כי על זה אתה מפעיל את הפילטר. אז זה ברור שלגמרי, שאם תסתכלי שברמת העין זה שונה לגמרי, אבל ברמת הפרספט אתה יודע לחלץ אחורה לפי הרפלקטנס, אוקיי? וזה קצת יותר מסובך מאשר להסביר את זה פה, ואני ממש לא זוכר בדיוק את הפרטים,
לא משנה,
אבל זה בפירוש יותר מתוחכם מה שדיברתי פה. זה לא ברמת הרטינה, זה ברמת הרטינה, הדברים הכי פשוטים, זאת אומרת, אוקיי, תראה, הנה אני
יכול... אותו מספר, אותו פרספשן, כאילו...
בדיוק, שיש לך כאילו 3 מספרים. יש לך 3 מספרים, וברגע שיש לך אותם 3 מספרים, אתה מקבל אותם 3 מספרים. כי זה נראה, אוקיי, זה מערכת נורא טריוויאלית, אוקיי, שכאילו וקטור שמורכב מ3 מספרים.
כן, בדיוק. עכשיו, אני חייבת לשאול אותך, כי בן זוג שלי הוא עובר צבעים, ותמיד אני כאילו מטרפת על זה, כאילו איך הוא רואה את העולם. סתם, לא ניכנס לזה עכשיו, אבל כאילו...
יש לו 2 פוטורצפטורים, הקלאסי, אני לא יודע בדיוק.
יש לו את כל הסוגים.
יש לו את כל הסוגים, אז אוקיי. אוקיי, אז הקלאסי הוא בדרך כלל עיוורון צבעים, שאתה, יש לך אחד הפוטורצפטורים לא מתפקד. אוקיי? ואז התוצאה
אחד הסוגים?
אחד הסוגים. כן, יש 3 סוגים של פוטורצפטורים.
הוא כן רואה, הוא יודע מה זה כחול, הוא יודע מה זה אדום, כאילו...
לא, ואז... אני לא יודע, צריך להביא אותה, יש כאלה מבחן של נשירה. אז בדרך כלל יש להם בעיה, כן, הוא יודע מה זה כחול, הוא יודע מה זה אדום, והוא לא יודע להבחין בין אדום וירוק.
נכון, בדיוק.
אוקיי? אז כאילו הבעיה היא שתחשבי רגע שבעצם, נגיד אם אחד הפוטורצפטורים הזה לא קיים. כאילו, יש לך 3 מספרים הרי שאתה מקבל. עכשיו, נכון? באופן נכון. אוקיי, עכשיו, אם אחד מהם לא קיים, אז הבעיה היא שאתה לא יודע להבחין. זאת אומרת, נגיד, למה הבעיה שבלילה אתה לא רואה צבע?
גם בלילה וגם רציתי להגיד בצלילה, שאתה צולל...
לא, בצבע אחרת, זה פיזיקה שלהם, כי האור בצבעים האדומים, לא. בצבעים האדומים הוא פשוט נבלע על ידי כאילו המים. אוקיי? אז כאילו, כשהאור נבלע, אז הוא לא מגיע. אוקיי, אז כאילו, את מקבלת רק את הכחולים, וככל שאת נכנסת יותר לעומק, בסוף אין לך בכלל לא, כן? כן, אבל יש בליעה ספציפית בצבע מסוים מאשר באור. כן, נכון. אז אותו דבר פה, תחשבי שאת מורידה את אחד בקומת הבליעה האחת הזו, היא לא קיימת. אוקיי? אז יש לך בעיה, כי את לא יודעת, זה כמו שאמרתי בלילה, את לא יודעת אם נתתי אור, נגיד, תסתכלת על הרוב. אז למה את לא רואה צבע? כי את לא יודעת להגיד האם קיבלתי פה, אני יכול לתת נגיד אור בתחום של 510 או ספר, מה שהמספר פה, כאילו, בעוצמה מסוימת, או בתחום, נגיד, הצהוב, בעוצמה יותר חזקה. תחשבי, אתה נותן בצהוב עוצמה חזקה, או בירוק עוצמה חלשה. בשני המקרים אני מקבל את אותו דבר. נכון, מבחינת הבליעה, זה המכפלה של עוצמת האור, כפול הפונקציה של הבליעה. נכון? כן. אז את מקבלת, זה אותו מספר, אז את לא יודעת להבחין בין האם זה אור צהוב בעוצמה חזקה, או אור ירוק בעוצמה יותר חלשה.
בגלל שחסרה לו קואורדינטה, נכון? כאילו
חסרה
קואורדינטה, אז הרבה יותר דברים הם...
אותו דבר, הם אקוויוולנטיים. והדרך שאנחנו מפרשים אותם זה שכאילו אנחנו לא... באמת, באור ירוק אנחנו נראה הכי טוב בלילה, ולכן באמת